giggle.
Okay, so we are going to prioritize finishing up this last bit of cardiac output, but I just wanted to let you know if we don't get to discussing it by the end of today, I'll send along email detailing stuff about finals if you've forgotten the various things I've been peppering in about that over the course of the semester, but basically our final is super short. It's only 16 questions, so it should not take you the full 2 hours that were scheduled, which means you'll have time to sit or retake of your choice directly after it, if you so choose. We have the last day of finals period, I think, so definitely all your other grades and stuff will be in. So you can also make that decision kind of on the spot after you take your final exam in series 4. So I'll send details about how that works. So we've got it all in writing for you, but that's kind of that squared away. Okay. So we're going to talk about this last little bit of cardiac output. Okay. So we were talking about how cardiac output is equivalent to our stroke volume times our heart rate. What we're trying to figure out is how much blood is your body getting per minute. And we know that to change our cardiac output, therefore we can change either stroke volume or heart rate or both, right? So we can do that with our sympathetic and parasympathetic nervous system, both of which can affect heart rate. We can also do that hormonally a little bit. So we talked about the sympathetic control of heart rate at the end of class on Monday. Basically, your sympathetic nervous system speeds up your heart rate, right? So speeds up your heart rate would increase cardiac output. These hormones do the same thing. And mostly on the same kind of pathway. We're going to click through it again right now, but we saw that our sympathetic nervous system has a connection to the SA node and the AV node, and it changes basically how fast we can depolarize the SA node, which is how it speeds up the firing of the SA node, because we're making it easier for action potentials to form. So epinephrine works basically the same as norepinephrine at those same places in the SA node. So it's going to speed up that SA node. It also speeds up a little bit how fast that action potential travels through the conduction pathway of the heart. Another hormone that's maybe a little more unexpected that it can have an effect on heart rate is glucagon. So if you remember glucagon from our endocrine discussions, that's our hormone of the post-absorptive state, right? So fun fact, during this period where you don't have a lot of freely available glucose in your bloodstream, it can be useful to get your blood moving around a little bit faster, make sure that you're getting that stuff. So glucagon can increase your heart rate a bit. Okay. All right. So when we're thinking about like integrating signals that all play a role on our heart rate, we know that with our autonomic branch of our nervous system, that kind of automatic homeostasis regulation piece, we never have just sympathetic branch active or just parasympathetic branch active, assuming they go to the same place. It's always like somewhere a little bit more of one predominating over the other. So that same thing is going to be true when we're thinking about heart rate at the SA node. So both the sympathetic and the parasympathetic neurons project to the SA node. Both of them have some level of activity. At rest, your parasympathetic nervous system is more active. So we'd say the parasympathetic nervous system predominates, which is why your heart rate is below 100 beats per minute, which is kind of our baseline activity of the SA node. Right. So both sympathetic neurons and parasympathetic neurons go to that heart rate. Sympathetics raise it, parasympathetics lower it. And our epinephrine works along with our sympathetic nervous system as it usually does, because that sympathetic pathway to the adrenal medulla is kind of looped in to this production of epinephrine that acts as a hormone. Right. So sympathetics, epinephrine raise, parasympathetics lower. So a reminder, this is our equation, right? So you can memorize the equation, or you can just remember the relationships, right? Our cardiac output, total amount of blood to the body. That means we gotta have the blood per heartbeat times the number of heartbeats. So we've been talking about changes to heart rate so far. So anytime we increase heart rate, increases cardiac output. Anytime we lower heart rate, lowers cardiac output. But we've got the other part of the equation too. So we've got the stroke volume as well. Now, this is going to be a little bit different than the heart rate part because we don't have parasympathetic innervation to the ventricular myocardium. Okay, so our vagus nerve doesn't touch the heart muscle. It only touches those pacemaker cells. It only touches that heart rate part. So when we're thinking about autonomic regulation of cardiac output, right, sympathetic can do both stroke volume and heart rate, but parasympathetics can only do the heart rate part. Okay. So when we're thinking about adjusting our stroke volume, there are a couple of ways that we can do that from kind of a physics perspective, right? So if you want to increase how much blood comes out when you squeeze the heart, right? So distally squeezing the heart, you've got blood ejected from the heart, right? You can adjust that by changing how hard the heart is squeezing. So that's our ventricular contractility part, right? We can also do that by changing a little bit about how the pressures in. So we know that the blood flow of the heart going from areas of high pressure to areas of low pressure. If the pressure gradient would send blood the wrong way, we put a valve there so the blood doesn't go backwards, right? That happens to be the case. That changing how much blood fills your heart in diastole, so that end diastolic volume has a little bit of a change on the amount of blood you're going to squeeze out as your heart contracts. And also the amount of pressure in your aorta, your afterload, because it changes basically how hard you have to fight against pressure in your arteries, in your vasculature, also can change stroke volume a little bit. All right. So if you want to join, I'm going to put a couple questions in a row as we start to talk about this stroke volume stuff. Keep it moving, but if you're not in here, just take down your answers on paper so you can still play along yourselves. Okay, so we're going to start out by predicting if we increase ventricular contractility, is that going to raise or lower our cardiac output? Right. Yeah. So if you squeeze the heart harder, that's the ventricular contractility, you're ultimately going to increase cardiac output because you're going to have more blood ejected every time your heart beats. So we're increasing stroke volume to increase our cardiac output here. All right. So our next piece of this, what if we increase our end diastolic volume? Do you think cardiac output is going to go up or down? All right. We'll take a look at this one, but this is actually also going to raise our cardiac output. So if we fill the heart with more blood, have a higher end diastolic volume, this is also going to end up resulting in higher stroke volume when we squeeze the heart. And that higher stroke volume ultimately will raise our cardiac output. All right, so the last piece, the afterload, which is pressure in the aorta. Is that going to raise or lower our cardiac output? Okay, yeah, that one's going to lower it, right? So for those three points we had on that stroke volume slide, right? The first two, right, are the ventricular contractility and our end diastolic volume. Both of those will end up raising cardiac output as they raise stroke volume. But this last one, the afterload, because it's a pressure you have to fight against, it's this pressure in the aorta. This is the one thing that's going to decrease our stroke volume and decrease our cardiac output. Basically, this makes it harder to push blood out of the heart. Right? And it's harder to push blood out of the heart, a little less blood manages to escape. Oh, they're going to be our takeaways at the end, but great job. Okay. So we're going to talk about the ventricular contractility part first. These are just some of the details. And so we know our end result of increasing ventricular contractility is going to be more contraction, means squeezing harder during systole, means we're going to squeeze more blood out, higher stroke volume, higher cardiac output. Okay. So how this happens is linked into the sympathetic nervous system. Okay. So we had our sympathetic control of the SA node before, right? So we had more action potentials, meaning faster heartbeat. This, we're now thinking about the sympathetic control of the heart muscle, the ventricular myocardium, so the muscle of the heart itself. Okay. So this works kind of similar, but we're in a different place. Right? So we have innervation to those muscle cells themselves. And at those cardiac muscle cells, what we're releasing is norepinephrine, right? So far, same as at the SA node. Right? We have adrenergic receptors here in our cardiac muscle in the ventricles. So we've got these beta-1 adrenergic receptors again, right? So they're still receiving that norepinephrine, and they're still activating that second messenger system centered around cyclic A&P. Okay? And what this does in a heart muscle cell, in the cardiac muscle, is that it's going to open our calcium channels. It's going to increase how much calcium gets released from the sarcoplasmic reticulum. So remember, when we're thinking about heart muscle, when we're thinking about muscle contracting in general, right? The way we have muscle contraction happen, we get an action potential come in, we release calcium, calcium binds to troponin, which moves aside tropomyosin, right? So this exposes myosin binding sites on actin, which means we can do cross-bridge cycling, right? So calcium is really the kind of trigger for allowing our cross-bridge cycling to happen, which is how we get sarcomeres sliding past, right? So what we're saying here is that norepinephrine coming from the sympathetic nervous system is ultimately putting more calcium in so we can have more cross-bridge cycling, right? More contraction of sarcomeres. The other thing that our second messenger system is telling our cardiac muscle cells is actually increasing a little bit how fast the myosin can break down ATP. So even the cycles themselves, those cross-bridge cycles themselves, I guess we usually have it in that direction, can happen a little faster because we can break down our energy a little faster. And it also increases how fast we can turn off these signals. So it pulls back calcium faster into the sarcoplasmic reticulum too. This is important because like if you just squeeze once really hard, but it still took you a long time to relax, right? You wouldn't get the overall effect of beating faster and harder at the same time, right? So we speed up our heart rate with that SA node activation. We increase our contraction with this increased calcium, But to get those two things to go together and actually increase cardiac output, we also have to make sure that we relax enough, fast enough to actually fill with blood in between those faster heartbeats. Okay, so in your version of the slide, you have a step-by-step walk through this whole process. It's not super important to me that you know all these steps in order. So to keep things a little lighter on this last lecture day of class, I'm just going to point out a couple of things for you to notice on here. All right, they're all things that we just mentioned in the text on that last slide. All right, so what we're ultimately doing is cross-bridge cycling, right? So this is that ultimate like contraction of the ventricles, right? Ultimately, we're squeezing the heart harder. This is our goal, right? So all we're looking at in this picture is that this is happening because we have norepinephrine, or we can already see that epinephrine, our hormonal effect, is going to be the same at this beta-1 receptor. Right, so here we see that beta-1 receptor, type of adrenergic receptor. We can see it activating cyclic A&P, right? And we can see that the effect here is a bunch of calcium stuff, right? So we're letting in more calcium. All right, so calcium activates cross-bridge cycling. So we're getting more calcium in from outside. We're getting more calcium out of our sarcoplasmic reticulum, right? Again, speeding up and increasing that cross-bridge cycling. And then we have that relaxation piece too. All right, so we speed up this reuptake of the calcium so that we can reset for our next heartbeat. That's all we're kind of like focusing on here. So the result is that if we were to look at like the twitch of our cardiac muscle, right? So over time, how much tension do we produce? Right? At rest, so when we don't have the sympathetic activity increased and in overdrive, right? We've got this curve, so kind of filling the whole space on the graph, right? So we got a heartbeat, kind of a gentle one. This is our ventricular contraction, gentle slope down back to rest, right? When we increase sympathetic activity, when we release this norepinephrine, trigger those beta-1 adrenergic receptors, make cyclic A&P, basically floods our cardiac muscle with more calcium and then take it away more quickly. Right. Here we can see the effect of that increased and more rapid calcium. Right. So we peak faster and we even peak higher. Right. So that's that increase in contractility. This happened both sooner than it usually would, and it produced more tension than it usually would with this extra calcium. And we can see that we come down from that peak faster too, right? So this slope coming down, this is that relaxation piece, right? So we're doing reuptake faster, we're sucking that calcium out of the cytosol faster so that we can reset, because we know that when the sympathetic nervous system is active, our next heartbeat is going to come a little sooner, right? So we want to be ready for a faster heartbeat as well. So parasympathetic nervous system does not go to the ventricle, so it does not have an effect, right? So when we're thinking about heart rate, both branches of the autonomic nervous system have an effect. You can speed up or slow down. But when we think about the ventricle, only the sympathetics have an effect, right? Because they're the only ones that go there. Parasympathetics not doing anything. Okay. Hormones do have an effect. They basically work just like the sympathetics do, right? So basically we have a lot of ways we can increase contraction, right? So as usual, epinephrine is working along with the sympathetic nervous system. When we took a peek at that beta-1 adrenergic receptor, we even saw it'll take epinephrine just the same as it'll take norepinephrine, right? So that would work basically exactly the same from the perspective of a cardiac muscle cell. And we have other hormones that can do similar things. So they wouldn't bind to that same beta-1 receptor, but overall they'd have the same effect of increasing contraction. So again, glucagon is on there, increasing force of contraction. Other hormones can potentially play a role here too. So some thyroid hormones, even insulin. If you have a lot of either of these hormones, you can also kind of have some effects on that force of contraction. That's just a little fun detail for us there, rather than a big thing to focus on. You do want to spend some time understanding this end diastolic volume. Right? This was the one that was a little trickier for us on the prediction. Okay. So when we think about end diastolic volume, we actually are partially thinking about something called Starling's law. And we're going to say of the heart, because we're going to have Starling showing up again in A&P 2 when we talk about like capillaries and kidneys and stuff. So the Starling's law, part 1, later on we'll have Starling's forces. Okay, what this is, basically, at the very end of discussing sarcomeres and some of our like muscle fiber stuff, we mentioned very briefly that muscles have an optimal length, right? So muscles have an optimal length, sarcomeres have an optimal length, right? When we just have just enough overlap that all our myosin and all our actin can kind of grab together, and we have enough space to fully contract without getting kind of blocked by the ends of the sarcomere, or without having to like reach too far to the actin and not be able to grab it, right? So that's true of sarcomeres in general, and that's true in our heart cells as well, just like it was in skeletal muscle. Now, it happens to be the case in your skeletal muscle, you usually like at rest, you're like at optimal length with a skeletal muscle, but in your heart at rest, you're not quite. Actually the case, but at rest, you're a little suboptimal. You're a little kind of too squished when your heart is relaxed. And if you fill a little bit more and your heart gets a little bigger, a little more stretched out, that actually moves you closer to the perfect length for your sarcomeres to be able to contract as much as possible. So this is what's happening when we see this change in contractility in response to an increase in end diastolic volume. Right? So end diastolic volume is happening. We have our heart filling with blood, right? So we've got our veins coming into our right atrium or into the left atrium if we're thinking about coming back from the lungs. Right? We've got blood filling the ventricles first as it's kind of just leaking and flowing down from the atrium, and then filling a little more as the atria contract, right? We're filling the ventricle with blood, right? So at the end of diastole, at the end of this relaxation period, we have the most possible blood we can have in the heart. That's when we would measure our end diastolic volume, right? And if you have a little extra blood, you had a little extra venous return, this means you're going to be a little closer to our optimal length of a sarcomere. If you're closer to your optimal length for the sarcomere, when you contract, it's a little stronger. When we contract a little stronger, this results in a little increase in stroke volume. Right? Squeeze harder, just like you squeeze your toothpaste really hard, all of a sudden you're going to shoot out a lot of toothpaste. Same thing happens if you squeeze a ventricle harder. So our kind of workflow here for ultimately increasing stroke volume, which would then increase cardiac output because it's part of our equation, right? Or part of our relationship, depending how we're thinking about it, right? Is we're thinking increase in venous return, so increase in veins, bringing blood back to the heart, increases that end diastolic volume. We're at optimal length, so this increases how strongly we can contract the ventricle. If we contract it more strongly, we're going to squeeze out more of the blood that's in there. So here's kind of the picture that we saw before, or at least saw kind of in some form. The optimal length piece, right? So here we're looking at two different sarcomeres, right? So here we can see that this one, a little more squished together, right? You don't have so much room to move, kind of, you can think of it, versus here, got a little more space, right? So what we're looking at here, if we read this chart the other way, right? So this chart has different labels on it than it would before because we're now being specific about the heart. Right, we're looking at the end diastolic volume on the x-axis. So this is how much blood filled your ventricle. Right, so if it's a little less, all right, I'll read up less. Right, along this curve, we'd see that the stroke volume is a little less, right? So lower end diastolic volume, lower stroke volume. Right, if we increase that, right, fill with a little more blood, read up to this curve, we can see that this slightly more stretched out sarcomere happens to be at more of an optimal length. So when it squeezes out more than our lower end diastolic volume. What you're looking at, this line, this curved teal line, that is the actual Starling curve. And the reason I'm pointing this out to you is that certain things can like shift the whole line. So I think we'll have some examples later, but we can see this line as a whole shift up, in which case you'd read it the same way, right? You'd just be reading this point and then this point, and you'd end up seeing the same relationship for end diastolic volume and increasing stroke volume, even if you had a curve that was a little higher than this. So here, so this is where we have like an interplay between our nerve affecting ventricular contractility and this relationship that we have between end diastolic volume and stroke volume. Okay. So remember, right, the end diastolic volume on that previous slide, right, we were saying if you go from this point on the curve to this point on the curve, right, you increase end diastolic volume and you also increase stroke volume, right? So that's one relationship we have along a curve. But we also know that nerves affect, specifically our sympathetic nerves, affect how hard the heart contracts. So basically this is just a layered effect, like it's additive, but not exactly like mathematically one-to-one additive, right? So all we're saying here is that if you increase sympathetic activity, this whole curve goes up, meaning any end diastolic volume, if you have more sympathetic activity, will result in higher stroke volume, right? But at that higher level of sympathetic activity, you could still make your end diastolic volume even higher and get that stroke volume even higher, right? That's all this is. So we have this relationship between stroke volume and end diastolic volume, and also sympathetic activity can still increase you further, but it would shift the whole curve. Right. We also have that lower down line. Just want to make sure that you're actually reading the label on it, right? So increase in sympathetic activity raises stroke volume. If we want to lower stroke volume, our parasympathetics can't do it because they don't go there, they don't have a connection. The way you would lower stroke volume is just by decreasing the sympathetic activity. So this is unusual, in which usually we see sympathetic activity going one way, parasympathetic activity going the other way. In this case, it's just more sympathetic activity versus less sympathetic. Okay. Cool. All right. So increases in end diastolic volume, increased stroke volume, increases in sympathetic activity, increased stroke volume. Another sort of way that we can think about end diastolic volume
is we can link that up to end diastolic pressure. So sometimes you'll see this word preload. The difference between our end diastolic volume and our end diastolic pressure is not something that I am super concerned that you can tell a big difference between, although as you go on to, clinical kind of classes, you may be paying attention to that stuff here. But basically, right, we're thinking about all of our filling, right, happens because of pressure gradients, right? So if you have less volume, that's the same as less pressure. If you have more volume, that's ultimately the same as more pressure at the end. So these are kind of both ways to be describing this increase in filling. When you have this larger end diastolic volume, right, this means this increase in filling can increase the amount of pressure that you first have in the atria, which means you fill the ventricles faster because you have higher pressure in the atrium, leading to lower pressure down in the ventricles. Another way you can affect this end diastolic volume is by affecting the pressure in the veins. We haven't really talked about venous pressure before, but basically, right, like that works the same as all our other pressure gradients did, right? A higher pressure in the vein, right, coming into the heart is gonna mean that you fill the heart faster because you have to go from a high pressure to a low pressure to fill the heart. Okay, so all those things are just kind of tied together. And if you were referring to it from this kind of pressure perspective, you might call it preload. Right? So that is a term that kind of shows up more in clinic in part because it makes it very obvious that it has the opposite effect from afterload. So we're going to talk about afterload specifically now, but we can just kind of link these together for ourselves. Okay. So for afterload, we're thinking about pressure in the aorta, right? So we're thinking, remember, heart, right? So our aorta coming out of the left ventricle, big and curvy, right? We know that in order to fill that aorta with blood, we have to have a higher pressure in the ventricle and a lower pressure in the aorta, right? We gotta work on this high-low gradient. So the problem with afterload, right? Okay. Basically, right, that as soon as you have a high pressure in the aorta and a lower pressure in the ventricle, right, you need that aortic semilunar valve to slam shut so that you don't have blood flowing backwards, right? So high pressure in the aorta while you're trying to eject blood is going to cut things short, right? So it's going to cut short our stroke volume. So that's what we want to know about afterload. Afterload is really the one factor on this whole list that we're going to be thinking of as decreasing our stroke volume, right? So increase sympathetics, you're going to increase stroke volume. Increase end diastolic volume, you're going to increase stroke volume. Increase the afterload, that's going to be our one exception. You have to fight against this pressure in the aorta. That'll decrease your stroke volume. All right. So, that's just a question that's not showing up. Okay. But basically, then we want to remember linking stuff up, right, to get to our total cardiac output. We're thinking about stroke volume. We're thinking about heart rate. So mostly we've been tracing through factors that increase our cardiac output, right? So we know sympathetics, increased stroke volume,
which means that they increase cardiac output. And they also increase our heart rate. Right? So the only way to lower cardiac output with our sympathetic nervous system would be if you're overall just decreasing your sympathetic nervous system, if you're turning it down a little bit. Parasympathetics don't have an effect on stroke volume, but they do decrease heart rate. So that's the way they can decrease our cardiac output. All right. So here we have a flow chart linking together our effects on stroke volume specifically. All right, so this is just another way to kind of visualize it for yourselves. Nothing here is different than what we've just talked about. All right, we know we are focusing in particular on the fact, the easiest one to understand is that increase in sympathetic activity, or epinephrine, our hormone associated with sympathetic activity, is going to increase contractility, right? Parasympathetics do nothing. Another effect that would likely be happening kind of at the same time would be an increase in end diastolic volume. A way in particular that would be happening is if your veins are sending back more blood more quickly, higher pressure. Right, so both those things would increase our stroke volume. Right. Afterload is the pressure in the arteries, so we have to fight against it. So if we're thinking about afterload in isolation, it would tend to decrease stroke volume. So what we'll notice on this chart is that a decrease in arterial pressure, so like a drop in pressure in the aorta, so like the opposite direction, would then mean it's easier to squeeze blood out to the body. So that would increase our stroke volume because we're going the opposite way. So this is just kind of summary together of these effects on stroke volume. Here's just a link to a video that I think summarizes some of this stuff. We don't need to click into that for today. Just wanted to make sure that we got an eye on the prize about what's high priority for our exam on Friday. So we do have spillover from our unit 3. So particularly what you want to remember from that, what's most important is remembering our muscle fiber types. So those like glycolytic fibers and oxidative fibers. You want to go through that, make sure you're up on that. As well as our forms of ATP generation. So, right, that glycolysis, oxidative phosphorylation, and now we know that there's also that on-tap energy in the form of creatinine phosphate. So we can link those up to like different types of exercise. We did that at the beginning of this kind of unit as well. So beyond that, when we're thinking about heart, we've got a lot of heart stuff. So you are expected to know it all. But if you're looking for what to particularly focus on, I would make extra sure that you know the steps of the cardiac cycle. So you want to know what order we have blood flowing through the heart and out to the aorta in. Right? You want to know how that links up to the steps of the cardiac cycle. You want to know how that links up to the pressures in the different chambers, as well as the state of the valves. And for the stuff that we covered today and on Monday, right, the way that you should be practicing that in particular is focusing on like our big picture point, which is how are you going to get more blood out to the body or less blood out to the body? So that's our cardiac output. So you want to be thinking about this stuff in terms of like always linking it back up to, would this increase our cardiac output? Would this decrease our cardiac output? And is it which of these two factors is it tied to, the heart rate part or the stroke volume? In the announcements, I posted, not like for points, you don't have to show it to me or do it, but I put a review slide together, a set of review slides together for you that you can make a copy of. You can drag and drop labeling parts of the heart for it. We've got some cycle stuff in there, some fiber stuff. It has a key links at the end. Also linked some little practice Google quizzes for you if you're looking for more stuff to study from. If not, don't worry about it. Okay, cool. So that is us for today. So we're taking exam for Friday. For our final exam, we got about a week to prepare for that. I'll in general be around during the week of finals if you want to stop by. I'll send out like a link where you can tell me that you're planning to stop by, so I don't go for a midday ski exactly when you were planning to swing by my office. And I'll send out for the final exam some like highlights of what we did in A&P 1 for each unit to kind of focus your studying on that, because we're going to be going ideally for like more big picture, most important topics rather than like little nitty gritty details.